So today we're going to focus on really just one part of the cell that we've discussed so far and how it works. And the part that we're going to look at in some depth today is the plasma membrane. Now notice the plasma membrane is also sometimes called the cell membrane. So two different names for the same thing. And if we look at living cells, all living cells are completely surrounded by a plasma membrane. And if you puncture the plasma membrane, if you poke a hole in it, it's kind of like poking a hole in a balloon. Instead of letting the air out, if you poke a hole in the plasma membrane, the cytoplasm and the organelles leak out, the cell dies. If it's a very tiny hole, sometimes it can be repaired before the cell dies, but usually the cell's a goner. So the plasma membrane is important. It keeps the cell intact, but even more than that, since the plasma membrane completely surrounds every cell, this means for anything on the outside to get into the cell, it has to pass through the plasma membrane. For anything on the inside of the cell in the cytoplasm to get out, it has to go through the plasma membrane. So the chemistry of the plasma membrane has a lot to do with how things get either into the cell or out of the cell, depending on where they're going. So we want to look first at the chemistry of the plasma membrane, and then we'll look at the different ways that things can get into and out of cells. So here's a section of a plasma membrane. Down here on the bottom is where the cytoplasm would be. One way we can tell this is because here are some of the cytoskeleton proteins. And the cytoskeleton is in the cytoplasm, so that's inside the cell. Now remember what we've said, the most common molecule in the cytoplasm is water. So there's a lot of water down here. Up on top of this plasma membrane drawing, this is where the external environment of the cell is, wherever the cell is living. And cells live in different environments, but cells are almost always found in environments that have some water in them. So there's also water up here. So in a sense, when we talk about controlling how things move into the cell or out of the cell, another way to think about this is keeping the water and everything in the water on the inside of the cell separate from the water and everything in the water on the outside of the cell. We don't want those two to mix. The chemistry of your cells, the types of molecules that we find there are very different than the molecules that we find outside your cells. And it's crucial that we maintain that difference. Usually when the chemistry of the inside of the cells becomes the same as the chemical on the outside of the cell, that's because the plasma membrane has been ruptured and the cell is dead. So we want to keep that in mind. So notice we see a lot of these little peach colored dots. And these peach colored dots represent the polar heads of those molecules that we've been talking about for a few days, the phospholipids. So these polar heads are hydrophilic, they mix with water. And the reason they're polar and hydrophilic, this is where the oxygens in the phosphate group of the phospholipid are found. This is where the oxygens from the carboxyl group of the fatty acids are found. This is where the oxygens from the hydroxyl functional groups of glycerol are found. So lots of bonds here between carbon and oxygen, between phosphorus and oxygen, and those are polar bonds. So we have a layer of phospholipids that is on the outside of the cell. Now remember that we have some little stringy things, two strings, attached to each polar head. And those two stringy things are the nonpolar tails. And this is what's left over from the two fatty acids that combined with glycerol. And they're nonpolar because if we look at their chemistry, all they have is carbon to carbon bonds and carbon to hydrogen bonds. And remember, carbon to carbon bonds are nonpolar, carbon to hydrogen bonds are nonpolar. Now, if we look, there's actually 2 layers of these phospholipids. So we can see a lot on this outer layer. We can just see a little bit down here. Notice these phospholipids are facing opposite directions. That's because we want the polar heads, these little peach-colored balls, we want those to be facing out against the water of the external environment, and we want them to be facing in against the water of the cytoplasm. So the membrane is referred to as a bilayer. Bi means 2. And we have 1 layer facing in, 1 layer facing out. Notice that all the nonpolar hydrophobic tails are in the middle of the plasma membrane's thickness. So the very outer part is polar to mix with the water in the outer environment. The very inner part is polar to mix with the water in the cytoplasm. Most of the thickness of the membrane is nonpolar and hydrophobic. And since we know nonpolar and hydrophobic things don't generally mix very well with polar things like water, this means that the water and everything that's in the water in the cytoplasm is trapped on the lower side. The water and everything that's in it in the external environment is trapped on the upper side of this membrane. So the main thing that the phospholipids do is really provide a barrier. They stop things from getting through. Now, there are a few things that can get through. Since most of the thickness of the plasma membrane is nonpolar or hydrophobic, things that are nonpolar can get through. Well, what type of molecules are nonpolar that living organisms deal with? O2, oxygen gas, is nonpolar. CO2, carbon dioxide, is nonpolar. Lipids are nonpolar. So oxygen, carbon dioxide, a lot of lipids can basically just kind of swim across the plasma membrane. We'll look at how they do this later. But those are about the only types of molecules that can get across the plasma membrane without some significant help. Now, the phospholipids make up about 90% of the molecules in the plasma membrane. So it's mostly phospholipids. Now, all these kind of light sky blue things are different proteins that are found in the plasma membrane. And there's a couple of ways to look at these proteins. Some of the proteins are actually kind of stuck in either the inner layer or the outer layer of the membrane. And these proteins that are in one of the layers, or some of them go all the way across both layers, these proteins that are in one or both of the layers of the membrane are called integral membrane proteins. Integral means part of. On the other hand, we have some proteins that are kind of floating. Here's one on the lower surface. I'm not sure if any of these are floating on the upper surface because I can't see their bottom, but in real life, there are some proteins that float on the top of these phospholipids. These are called peripheral membrane proteins, and peripheral means to the side. So they're not inside the membrane like the integral proteins, they're more to the side, to the top or bottom layer. And membranes have both. Now, some of the membranes are basically like a pipe. They're a hollow tube that run all the way from the inside of the cell to the outside. And these are called protein channels. Now, remember we said most things can't just go through the plasma membrane. They have to, get some help. These protein channels are one type of help that they can get. Protein channels allow materials to move from one side of the membrane to the other. But remember what we've been saying about proteins every chance we get. We're going to say it again. Proteins work by their three-dimensional shape. And the only things that can move through these protein channels are things that have the correct shape, which really means things that have the correct chemistry. So these protein channels don't necessarily just let anything through. They're very specific. There are different protein channels for different substances. So it's not a one-size-fits-all situation. There are also some proteins, some of these integral proteins, that work kind of like, I guess I would compare them to a handoff in football. We have some of these integral proteins like this triangle-shaped one here, and they may pick up something on the inside, and then they swivel around and they drop it off on the outside. So there are a lot of different ways that proteins can help materials move from one side of the membrane to the other. But transporting chemicals through the membrane isn't the only thing that these proteins do. Some of the proteins on the outer surface, now notice this is only proteins on the outer surface. This isn't true about proteins on the inner surface of the membrane. But some of the proteins on the outer surface work as recognition molecules, or ID tags, you could think of them. And they tell, if we're looking at a person, they tell your immune system two things. They tell you, first of all, that this is your cell. And they tell you, second of all, what type of cell it is. Every cell in the body doesn't have the same recognition proteins. So some of these proteins are ID tags. Now, some of the proteins that are ID tags are specialized. You can see that some of these proteins, there's one up here at the top that we can see pretty clearly, that has a branch of little blobs coming off of it. These little blobs are carbohydrates. And typically it's somewhere between about 5 and 15 monosaccharides bonded together. So it's a type of oligosaccharide. It's usually branched like we see here, so it's usually not a straight chain. And these molecules are very important for recognition. This is one of the main things that our immune system checks. Now, notice that this molecule, this carbohydrate, is attached to the outer surface of the protein. And if we look at these proteins, think back to what we said a moment ago about our phospholipids, the part of the protein that sticks out of the membrane, whether we're looking at the outer surface or the inner surface, the part of the membrane that sticks out is polar. The part of the protein that's in the middle, if we're looking at one of these channels, one of these proteins that goes through the center of the cell, that section of the protein is nonpolar, it's hydrophobic. So just like with the phospholipids, the polar part of the membrane is towards the outside. If we're in the outer layer, it's towards the inside if we're in the inner layer. Now notice we only have these carbohydrates attached to proteins in the outer layer. And they're always attached on the very outside, so on the polar part of the protein. And remember, when we say polar, we could also be saying that it's the hydrophilic part of the protein. So when we look at this molecule, since it has part of it that's protein and part of it that's carbohydrate, this is called a glycoprotein. Now, glyco as a prefix always refers to carbohydrates. Remember that one of the carbohydrates we talked about was glycogen. So same kind of basic root to the word. So this is a hint that this molecule is part carbohydrate, part protein. As I say, it's used a lot in recognition. This is one of the main things our immune system checks. Now notice we also have some chains of carbohydrates coming off the polar heads of the phospholipids. And this is called a glycolipid. And again, this is typically a short branch chain made up of somewhere between about 5 and 15 monosaccharides bonded together. And again, this is used for cell-to-cell recognition. Now, if we look at the glycoprotein and the glycolipids combined, they make up something called the glycocalyx. And this glycocalyx, if we look at the outside of cells, forms a kind of almost like a fuzzy covering, because we have these little sharp molecules that stick up just a very short distance outside the plasma membrane. Remember, they're never inside the plasma membrane. So when we look at these, what we typically see is that these glycoproteins and glycolipids don't only work in cell-to-cell recognition. They also work in what's called cell adhesion. Cells usually stick to their neighboring cells. So some of these glycocalyx molecules form something called CAM, cellular adhesion molecules. They help molecules stick to each other. They can also provide a kind of a protection. If we look at the glycocalyx on prokaryotic cells, like we see in bacteria, the glycocalyx can stop some antibiotics from getting into the cell. Now, remember what we've said, the plasma membrane is really the outermost part of a cell that we consider living. So if a bacteria cell can kind of catch antibiotics on these glycoproteins and glycolipids, that means the antibiotics never really get down to the living part of the cell. So we've got phospholipids, we've got proteins, and proteins, we said phospholipids were about 90% of the plasma membrane. Proteins are about the other 10%. So mostly this is phospholipids and proteins, along with a few modified phospholipids, the glycolipids, and a few modified proteins, the glycoproteins. But if we look at an animal cell, there's one other type of molecule that we find in the plasma membrane. Now, if we were looking at say a bacteria cell or a plant cell or a fungus cell, on the outside part of the plasma membrane, those cells have a cell wall. And whereas the plasma membrane is, it's phospholipids. So this is kind of like, it's very flexible. It's very fluid. It can move around. Cell walls aren't. Cell walls are like the walls of a building. They're very thick and stiff and rigid. They don't have any give to them. So the cell wall kind of anchors all these molecules in place. Animal cells don't have a cell wall. So we have to have something so that all these molecules don't move around too much, because normally they can kind of float around, kind of like droplets of oil in a frying pan. And the molecule that animal cells have in their plasma membrane that other cells don't are these little yellow hexagons and pentagons. This represents a cholesterol molecule. Now, cholesterol is very nonpolar. So cholesterol is pretty much completely in this nonpolar hydrophobic region in the middle of the plasma membrane. And when we look at cholesterol, it's so strongly nonpolar, it doesn't move around very much. So when we see a cholesterol molecule, it kind of anchors all the phospholipid molecules around it in one spot so that they don't really move very much. So we mostly hear about cholesterol being a bad thing because if you have too much cholesterol, it can lead to heart disease and some other problems. But you actually have to have some cholesterol. If you could magically snap your fingers and remove all the cholesterol from your body, you'd die very quickly because all your cells would fall apart without this cholesterol to stabilize the plasma membranes. And sometimes when people go on very low cholesterol diets, they actually suffer from other health diseases, other health problems. So these are the main molecules that we find in the plasma membrane. So we want to keep these in mind when we're thinking about how things get into the cell and how things get out of the cell. So again, to summarize, what do phospholipids do? Mostly stop stuff from getting in or out. What do proteins do? Well, proteins are the specialists that move things into the cell or move things out of the cell. Proteins can also help in cell-to-cell recognition. Proteins act as receptors. They pick up things from the environment. You could think of them like the space station, where there's a loading dock where the next space capsule attaches. So, and then finally, we've got our cholesterol if we're talking about animal cells. If we're talking about plant cells, no cholesterol. So you do have to be careful when you shop, because I'm not sure if they still have it on their label, but there used to be a product, I know the product is still made, it's called Mazola corn oil. And Mazola corn oil has something on their label that says 100% corn oil. And then they have a big banner that says contains no cholesterol. Well, of course it contains no cholesterol. It's 100% corn oil. There is no cholesterol in corn oil. The cheapest generic corn oil you can buy, if it's 100% corn oil, doesn't have any cholesterol. They didn't remove all the cholesterol in Mazola. It wasn't there to start with. So it's not really a lie in their advertising. It's just irrelevant. What if they had a big banner on Mazola corn oil and it said contains no automobile tires? Yeah, so there aren't automobile tires in corn oil. What does it matter? So when we think about the plasma membrane, we want to think about this chemistry in terms of looking at how things get in and out of the cell. And the first thing we want to talk about is a process called diffusion. And diffusion is a physical, passive process. Now, when we say that a way that things move across the plasma membrane is a physical process, we mean that it happens just because of the laws of chemistry and physics. It has nothing to do with how cells work. And in fact, diffusion is used in a lot of industrial processes, because diffusion isn't a biological process. We don't need a living cell for diffusion. We don't need a cell at all. So there are a lot of industrial processes that use diffusion. And it's passive. And passive means that it doesn't need any energy. If you just sit here and do nothing, diffusion will happen. So here's an example of diffusion. We've got our lipid bilayer, in other words, a plasma membrane, and it's this kind of this light greenish line. We have some sort of chemical, could be atoms, could be molecules, could be ions. And to start with, they're all on one side of the membrane. But here's the thing, chemical particles, atoms, molecules, ions, are constantly moving. They're bouncing around. And as they bounce around randomly. You know, if you think of the room you're in as being a three-dimensional space, there are some of these particles bouncing to the left, some to the right. Some of them are bouncing in front of you, some are bouncing back behind you. Some are bouncing up, some are bouncing down. So they're going in all different directions completely randomly. And they're going at weird angles. They're not going straight up or straight down. They might be going at a 60-degree angle or something. As they move, even though they move completely randomly, we see a pattern because of this random movement. And the pattern is when we have particles and the particles are free to move, they're free to diffuse. They always diffuse from where they're at a high concentration, from where there are a lot of them in a small area, to where they're at a low concentration, where there are less of them. And they keep diffusing, and after enough diffusion, the concentrations are equal everywhere. There are just as many chemical particles on the top side of this lipid bilayer as there are on the bottom side of this lipid bilayer. Now, when we look at this final situation, we have something that's referred to as a dynamic equilibrium. An equilibrium, so a dynamic equilibrium, an equilibrium means things are equal. So what are equal? The concentrations of these little bluish purple dots, whatever they represent. But it's a dynamic equilibrium. What does dynamic mean? Well, dynamic means that all these individual molecules, maybe they're carbon dioxide molecules, all these carbon dioxide molecules are still moving randomly. But once we get to a dynamic equilibrium, concentrations don't change. Why? Because on average, for every molecule that's moving up across the membrane, there's another molecule up here that's moving down below the membrane. So the individual particles moving back and forth kind of cancel each other out. So dynamic refers to the fact that individual particles are constantly in motion still. Equilibrium refers to the fact that things are even. Now, you've all experienced diffusion. At some point, I bet you've spilled some kind of cleaning agent, maybe something with ammonia in it that has a fairly strong smell, or maybe you've spilled a perfume or a cologne. And when you first spill that substance, all the smell is concentrated right around the area of the spill. But as time passes, what happens is those molecules diffuse and the smell spreads out. So you might have a smell that starts out concentrated in one corner of the room, and it gradually spreads out throughout the whole room. Years ago, we did a great experiment when we were on campus. We got a skunk, and we put the skunk up in the front of the room under the teacher's desk. And when I lecture, when we're on campus, I don't usually sit down. I walk around the classroom. And every time I walk back behind the teacher's desk, I just gave the skunk's cage a little bit of a kick. And when you do that and the skunks become upset, they produce a chemical called beta-mercaptan. And if you've ever driven down a road where there was a dead skunk in the road, beta-mercaptan has a very distinctive smell. And so the skunks started making beta-mercaptan. At first, all the beta-mercaptan was basically underneath my desk in that little spot where you put your legs when you sit down at a desk. But as time progressed, the smell spread out. And first it spread to the, kind of the first row of seats, because diffusion happens, over areas. And the people in the front row started making weird faces and scrunching up their noses, and, nobody knew what was wrong with them. And then eventually the smell spread throughout the entire lab. So it was a really good demonstration of diffusion. Everybody remembered diffusion pretty well that semester. Now there's a special type of diffusion that can occur as well. Diffusion is the movement of anything. We could be talking about sodium ions, we could be talking about carbon dioxide, we could be talking about water molecules, all sorts of possibilities. And notice when diffusion happens, if the temperature is higher, diffusion is faster. If there's a low temperature, diffusion is slower. If the molecules are big, they diffuse more slowly. If they're small, they diffuse more rapidly. The bigger the difference between the areas that diffusion is happening between, the bigger the difference, the faster diffusion is. So here in the very left, diffusion would be occurring very rapidly. When we get over here in the middle picture, diffusion would be slowing down a little bit. Why? Because there's not as big a difference between the concentration above the lipid bilayer and below the lipid bilayer. So diffusion isn't always exactly the same speed. Sometimes it's a little bit quicker, sometimes it's a little bit slower. Gases diffuse faster than liquids. Liquids diffuse faster than solids. Now, whenever we talk about something, kind of a special example of something, think about what we've been pointing out all semester. What's the most common molecule in living things? Water. So the special type of diffusion we want to talk about is diffusion of water. And when we talk about diffusion of water, We're talking about a process called osmosis that you might have heard of. Now, osmosis is a type of diffusion. So remember what we said about diffusion a minute ago. Diffusion is a process that's a physical process, so it doesn't need a living cell, and it's passive. A cell doesn't have to use any energy. Now, diffusion still happens across a plasma membrane, even though it doesn't need a living cell. And osmosis can happen across a plasma membrane as well. So let's look at three different situations. Now, the cells that we're looking at here represent red blood cells in your bloodstream. So let's look first at an isotonic solution. Now, the reason that we want to look at this first is this is the normal shape of red blood cells. They would look the same on the other side. So they've got a little indentation in the middle on both sides. And this is what we see if we put red blood cells in an isotonic solution. Now, if we look at the water inside the cell, inside the cytoplasm, there's some water, and then there are some things dissolved in the water. Isotonic, remember we've talked about isotopes and isomers. Iso means the same. And isotonic has the same concentration of dissolved substances as the cell does. So this means that if anything moves into the cell by diffusion, it will also move out of the cell by diffusion. Or if water moves into the cell by osmosis, water will also move out of the cell by osmosis. So in an isotonic solution, they're showing the movement of water by these blue arrows. And the key thing to notice about these arrows is that they're both about the same size. And that's because the same amount of water is moving into the cell by osmosis as is moving out of the cell by osmosis. So because the same amount of water is moving into the cell and out of the cell, it doesn't change the cell at all. But now let's look at this diagram on the left that refers to a hypertonic solution. Well, when you see hypertonic, think of a child who's hyperactive. A child who's hyperactive is more active than usual. Hypertonic solution has more chemicals, more solutes dissolved in the water than is usual for the cell. So this basically means that we're putting these cells, keep in mind, these are red blood cells that started out with this normal shape. We're putting these red blood cells in very salty water. So think about if you've ever been to an ocean and you've gotten a mouthful of salt water while you were in the ocean. That water tastes very salty to you. So if we put cells in very salty water, cells that have a higher concentration, water that has a higher concentration of salt than the cells, what we see is the cells gain salt and they lose water. So notice this diagram shows a little arrow going into the cell. Just a little water is being gained by osmosis, but it shows a bigger arrow going out of the cell. More water is being lost. So what happens to the cell? Well, the cell kind of shrivels up like a balloon that you've let some of the air out of. We say that the cell crenates when it does this. Now, when the cell shrivels up like this, if it doesn't lose too much water, the cell can still survive if we add some water, if we rehydrate the cell. We would like, when we treat you in most medical procedures, to give you something that's isotonic, so your cells don't shrivel up. But this shriveling up is what we see, for instance, if you drink salt water. If you would drink salt water because you're thirsty, the water that you drink that's in your stomach and your small intestine is so salty, it actually pulls water out of your cells into your small intestine and into your stomach. So if you drink salt water, what happens? Well, you're going to be more hydrated after you drink salty water than before you drink it. If you lose enough water this way, you may have diarrhea, because if you've got salt water in your digestive system, you can't really reabsorb that water very well. Now let's look at the opposite situation though. Let's look at something called a hypotonic solution. And when you see this, think of a hypodermic needle. A hypodermic needle goes underneath your dermis. It goes below your dermis, which is one of the layers in your skin. So something that's hypotonic has a lower level of things dissolved in the water. The ultimate hypotonic solution would be just pure water, 100% water. Now, since there are some chemicals dissolved in the water of your cells, if you put red blood cells, and keep in mind this also applies to other types of cells, if you put red blood cells in a hypotonic solution, they lose a little bit of water, but notice there's a bigger arrow coming in. They gain more water. So it's kind of like blowing air into a balloon. The cells swell up, and if they swell up too much, they can actually burst like a balloon that you put too much air into. There have been several cases of nurses and doctors who have been convicted of killing patients. And how did they kill the patients? The patients were receiving an IV, and what did they do? They put pure water into the IV. So pure water is going into their blood vessels, and when it gets into their blood vessels, the red blood cells absorb water, swell up, and burst. And when this happens, keep in mind the red blood cells have a protein in them called hemoglobin, and hemoglobin picks up oxygen in your lungs, drops it off at other locations in your body. So if your red blood cells start bursting, you can't carry enough oxygen to your cells. So you could be sitting in a room like the room that you're in now, and you could actually suffocate, because suffocation just means that your cells aren't getting enough oxygen. So there, as I say, there have been a couple of cases where medical professionals have been convicted for doing this to patients. So when we look at diffusion and osmosis, they're both physical processes. They don't need a living cell, so they can be used in industry. They're both passive processes. They don't need any energy. So if we can use them in industry, it's beneficial because it's a cheap process. It doesn't necessarily require any energy. Now, the other processes that we're going to talk about are biological processes. And we know that biology refers to living. Biological processes need a living cell. So diffusion, osmosis, no. The other processes we're going to talk about yet. Now, we're going to talk about a passive biological process called facilitated diffusion. And the fact that it's called facilitated diffusion gives you a hint that it's passive. Because remember, diffusion was passive, and osmosis, which is a special type of diffusion. Osmosis is just diffusion of water. That's passive. And we're going to talk about several biological processes that are called active transport. And as the name suggests, in active transport, the cell has to be active, which means the cell has to use energy. So let's think first about facilitated diffusion. So in regular diffusion, we have some kind of particle, and that particle can just pass through the plasma membrane. It can move from one side of the plasma membrane to the other side. Now, the big thing that's a benefit of diffusion is diffusion doesn't require the cell to use any energy. But there's also a downside to diffusion. Suppose you're a cell and you need 100 of these little purple molecules. In diffusion, every molecule moves independently. So when a molecule happens to move across the plasma membrane into the cell and you can grab it, it comes in one molecule at a time. And remember that in diffusion, movement is random. So think about if you were, at the grocery store or at a mall, if you were someplace and you got home randomly. That would be like coming to the first corner, flipping a coin. Heads, you go right, tails, you go left. If it stands on edge, you go straight ahead. You could get home that way, but it's going to take forever. So diffusion has one main disadvantage. Well, two, I guess. It's really slow. Now, the other thing is, keep in mind, ultimately, diffusion can only move things from the side of the membrane where they're at a high concentration to where they're at a low concentration. In facilitated diffusion, we have a protein. That's what these big purple blobs are. And the protein might work like a channel, or it might attach to a molecule and then kind of squeeze the molecule through to the other side, kind of like squeezing a tube of toothpaste. Notice that the proteins, as we've been talking about, are specific. They work according to their shape. So they will be able to help some molecules move across the membrane, but not all molecules. Now, this is facilitated diffusion. So this means it kind of works like diffusion did. You facilitate something, you make it easier. So this still works like diffusion. It only moves molecules from the side where they're at a high concentration to the side where they're at a low concentration. It doesn't require any energy use on the cell's part. So why bother? Well, it turns out facilitated diffusion has one big advantage over diffusion. Facilitated diffusion is faster than plain diffusion. Why? Because you have a protein that's picking molecules up on the side where they're at a high concentration, pushing them over to the side where they're at a low concentration. So facilitated diffusion is faster than regular diffusion. Otherwise, it's about the same. Now, in active transport, we typically have some type of protein involved. And again, the protein moves molecules across the membrane, but here it takes energy. Remember that ATP is the molecule that cells use for energy. So it takes energy to move molecules across the membrane. And when we move things across the membrane, active transport, since we have a protein helping the movement, has two big advantages compared to diffusion. It's faster because just like facilitated diffusion, we have a protein helping to move things, but the biggest advantage is we can move chemicals. Look at our little kind of our yellowish gold diamonds here. We can move our molecules from where they're at a low concentration. Notice there's only 2 out here, to where they're at a high concentration. There's 5 in here. So we can move things in the opposite direction that diffusion moves things. So this can be very useful. So when we talk about moving things from low to high concentration, this is sometimes called against the concentration or against the concentration gradient. And why does it take energy? Well, it's sometimes compared to pushing something uphill. Diffusion is like something rolling downhill. Yeah, you don't really have to do much. It rolls downhill just because of gravity. This is pushing something uphill. That doesn't happen in nature. So one example of this is the CFTR channel. which is found in cells. And normally it transports chlorine ions out of the cell. If something goes wrong and it doesn't work, you get a disease called cystic fibrosis, because all those chlorine ions attract water and you get a mucus around your cells. One of the most important types of active transport is something called the sodium-potassium pump. Cells have a lot of potassium in them. They don't have very much sodium, but out around the cells in your blood or the other fluids surrounding cells, there's a lot of sodium and not very much potassium. So the sodium pump pumps 2 potassium into the cell, and at the same time, it pumps 3 sodium out of the cell. Kind of works like a revolving door. When the potassium is going in, the sodium is pushed out. But if you think about a revolving doors take energy. They don't just turn by themselves. You have to push them, or some stores have automatic revolving doors, but then there's a motor doing the pushing. So something is pushing those doors. So that's why this is a type of active transport. Remember what we said, active transport needs energy. Now, the final ways that we can get things into a cell or out of a cell, as the case may be, is through some processes called endocytosis and exocytosis. Endocytosis refers to moving a lot of material into a cell all at once. Remember, we said one problem before was, you couldn't only get one molecule at a time by diffusion. Exocytosis is a way of eliminating a large amount of material from the cell all at once. So what happens in endocytosis? So we have some chemicals in the external environment. So those are these little purple blobs. And basically the membrane pinches in, and as it pinches in, it forms a little pouch or a pocket. And then this little pocket seals off. And when it seals off, we trap whatever those little purple molecules are inside the cell. Now, if most of the material that's brought into the cell this way is solid, this is a process called phagocytosis. Phago means eat. Cytosis is just a cell process. If most of the material that's brought in is liquid, it's pinocytosis. And we sometimes even talk about receptor-mediated endocytosis, where something has to attach to receptors located in the plasma membrane in this pocket area. Exocytosis is essentially like rewinding a film of endocytosis. We have some type of waste product in the cell. It's surrounded by a little vesicle or vacuole, which is basically a little membrane sac. This vesicle or vacuole moves to the plasma membrane and fuses with it. And then the plasma membrane kind of pinches back. And when it pinches back, now all this material is outside the cell. So let's look at a short video of endocytosis and exocytosis. Whoops. And I think it would help if I shared the right screen. Okay, you should be able to see a YouTube page now, I hope. Let me know if you don't.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein cladrine coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
Thank you for watching this video.
Are there any questions about the plasma membrane or how things get into or out of a cell? And keep in mind that all these processes are going on simultaneously. Cells are always exchanging materials with their environment, some more than others, but all of these processes are occurring at different parts along the plasma membrane.
I'm kind of confused.
Well, I would say watch the video again and look at the book and then check and see. And if you want to talk about it, send me a note. We'll meet and we can discuss it further. And unless you have a specific question now, then I'd be happy to try to answer it if I can.
I'll just watch a video again and look at the book.
Okay. I am going to post some homework that will be due when we come back next week, asking you about different parts of the cell. Keep in mind that the answers to all these questions will be some of the organelles that we talked about when we were discussing the cell. So if you go back and look at the notes over the prokaryotic cells and the eukaryotic cells, that's where you'll find the answers to these, or if you view the videos again. So this will be due next week when we come back, so that should be posted later this afternoon. Are there any questions? Okay, if there aren't any questions, I'm going to stop recording.